Fragile X syndrome is the most common single gene cause of a person developing autism and the most common reason for intellectual disability in males. The disease is typically characterized by mild to severe cognitive impairments, which are less severe in females, an elongated face, prominent ears, and post-pubertal macroorchidism, which is large testes. Severe obesity is associated with a phenotype of the disease, as well as hyperphagia, which is excessive eating, and hypogonadism, which is delayed puberty. Fragile X syndrome is caused by the expansion of the Trinucleotide CGG repeat on the promoter region of the gene FMR1, which stands for Fragile Mental Retardation 1 gene. This gene is on the X chromosome. As a result of this, the affected promoter region commonly becomes methylated, which is to do with gene expression and control. And the FMR1 gene gets turned off and instead of on, and the FMRP, which is the protein associated with the gene, is not produced. Methylation leads to a deficiency or complete absence of the protein. Mutation of the gene is found in around 1 in every 2,000 males and 1 in 259 females. However, the incidence of the disease occurring in males is 1 in 3,600 and 1 in 4,006,000 for females. Females are not as adversely affected because they have 2 X chromosomes. The FMR1 gene normally contains between 6 and 55 repeats of the CGG codon. In sufferers of Fragile X syndrome, it can be anywhere between 230 to 4,000 repeats. This repeat expansion is what causes the methylation of that part of DNA, which silences expression of the FMR1 protein. Molecules of RNA that have the expanded CGG codon initiate a cascade of events that conclude in the central nervous system. RNAs containing expanded repeats interact with different binding proteins and produce disease, which is a common characteristic of an expansion repeat disease. An FRM1 gene that is turned off or methylated as a result of more than 200 repeats of the CGG codon will not produce the protein that is required for normal neurological development, which leads to mental retardation in a person.
Hi, I'm talking today about Friedreich's ataxia. A background to this disease is that it's autosomal recessive, which means you get it from both your mother and your father. 4 in every 100,000 people are affected by it, and it is a genetic disease, which means you have a higher chance of getting Friedreich's ataxia if you have a family history of it. This disease wears away the parts of your brain and spinal cord that control coordination, muscle movement, and some sensory contractions. The symptoms include abnormal speech, changes in vision, mainly color vision, a decreased ability to feel vibrations in lower limbs, foot problems, for example hammer toe and high arches, hearing loss in around 10% of the patients, jerky eye movements, loss of coordination and balance, muscle weakness, like no leg reflexes, diabetes, which is a main symptom, and heart disease, which is also a main symptom, and this leads to heart failure. The muscle problems can lead to changes in the spine that can result in scoliosis, and this disease starts in childhood, before puberty. Tests to detect Friedreich's ataxia include X-rays of the head, chest or spine, a CT or MRI of the head, and an ECG or EMG or electrophysiological studies of the person. The mutations that causes this disease is a mutation in a gene called frataxin, which is located on chromosome 9. This mutation causes the body to produce too much of the DNA trinucleotide repeat GAA. Normally, the body has around 8 to 30 copies of GAA. However, in individuals affected with Friedreich's ataxia, they can have up to 1,000. This is called a triplet repeat expansion. The mechanism behind this disease is that the frataxin gene is found in the energy-producing parts of the mitochondria. When it mutates and is therefore unable to function properly, therefore the gene can no longer be correctly transcribed onto the messenger RNA. It is now blocked at elongation. This therefore means it can't produce energy like normal. This particularly affects the peripheral nerves, spinal cord, brain, and muscle cells. Because of the frataxin's inability to function normally, toxins build up within the mitochondria, leading to oxidative stress and increased iron levels. When the increased amount of iron reacts with oxygen, it forms free radicals which destroy cells and harm the body. It is believed that the nervous system, heart, and pancreas may be particularly susceptible to damage from free radicals, because once certain cells in these tissues are destroyed, they cannot be replaced. Currently, there are no known treatments for Friedreich's ataxia. However, the symptoms of this disease can be treated very effectively. Diabetes and heart problems, which are the main symptoms, can be treated with medications. Orthopedic problems, such as foot deformities, can be treated with braces or surgery. And physical therapy is also an option to extend the use of arms and legs. Hypothetically, if an iron-absorbing substance was ingested, it could absorb the excess iron in the mitochondria, therefore not allowing it to react with the oxygen and form free radicals.
Spinal and bulbar muscular atrophy is a hereditary neuromuscular disorder of the gradual degeneration of motor neurons. It causes a diverse range of symptoms in which affect sufferers' mental, physical, and physiological facets. Primary symptoms are characterized as muscle weakness and atrophy. It usually begins in adulthood, occurring predominantly in men. Approximately 1 in 50,000 males are affected. This ruthless disease is caused by an expansion of a trinucleotide repeat within the AR gene. Its onset occurs between 20 to 40 years of age, dependent on the severity of the mutation and the physical and physiological condition of the sufferer. Symptoms experienced by patients are wasting of the muscles in the arms and legs, cramping, loss of physical control, which is potentially dangerous due to lack of control, and tendencies to fall or collapse. Progressive development is also experienced with the loss of speech and the ability to swallow. Twitches may develop depending on the individual, as well as the development of breast and infertility. Mutations. Spinal and bulbar muscular atrophy is inherited by an X-linked recessive pattern. The gene is located on the X chromosome, which females are deemed carriers, although only mild symptoms are present in comparison to those experienced by men. The DNA segment of a sufferer undergoes mutation in which a CAG triplet repeat is irregularly expanded. In a healthy regular individual, the usual segment is repeated 36 times. However, in a sufferer, the nucleotide is repeated 38 to 60 times more. The mutated segment may ultimately become 2 to 3 times its usual size. Mechanisms. The change in the expanded CAG triplet repeat affects the structure of the protein synthesized by the AR gene. This therefore obstructs the regular function of the motor neurons within the brain and spinal cord. The nerve cells gradually die, thus leading to muscular atrophy and related symptoms. Individuals with larger amounts of CAG repeats tend to develop symptoms at an earlier age, relative to those whom have less. Suggested therapeutic treatments. There is no current cure for the disease, although management of symptoms are available, such as physical therapy, to decelerate atrophy. Removal of the undesirable CAG trinucleotides and extraction from their genes would be the preferred solution. However, no clinical method has yet been developed to cure the disease. In regards to muscular treatment, trials have been tested for the vivo activation of SMN2 by valproic acid. A clinical study of 7 patients, 6 of 7 experienced relative quick onsets to the treatment. Results found that an increase of 16% in muscle strength previous to treatment, which was sustained after a year.
Spinocerebellar ataxia type 1, also known as SCA1, is an autosomal dominant disorder. This means that approximately 50% of offspring also get the disease from either carrier or affected parent. This disorder is caused by a DNA CAG trinucleotide repeat on chromosome 6p22, a mutation of the ATXN1 gene. This gene codes for the ataxin-1 protein, and its normal cell function is unknown. When mutated, the three-dimensional shape of the protein is different to that of a normal protein and causes it to stop functioning normally. These mutated proteins aggregate together and collect in the brain and in the spinal cord. It predominantly affects the cerebellum, which is located at the base of the brain. The cerebellum coordinates movement and is very sensitive to any changes. Approximately 2 in 100,000 people worldwide are affected. Most people with the disorder start to notice symptoms in early adulthood. Generally, the earlier the symptoms appear, the worse the disorder affects them. People typically survive 10 to 20 years after symptoms first appear. The first symptom to appear is problems with coordination and balance. Others include speech and swallowing difficulties, weakness in the muscles that control eye movement, resulting in involuntary eye movements, and difficulty processing, learning, and remembering information. Over time, people may develop numbness, tingling, or pain in their arms and legs, uncontrolled muscle tensing, muscle wasting, and muscle twitches. The diagnosis of SCA1 is determined through molecular genetic testing to detect an abnormal CAG trinucleotide repeat expansion in ATXN1. Affected individuals have alleles with 39 to 91 CAG trinucleotide repeats. Such testing detects 100% of cases and is available in clinical laboratories. As there are no current cures for SCA1 yet, management policies can be put in place to help people live easier lives. These include canes and walkers to help prevent falls, modification of the home with grab bars, raised toilet seats, and ramps for motorized chairs, speech therapy and communication devices, as well as weighted eating utensils and dressing hooks to help maintain independence. It is also a good idea to stay away from alcohol and medications that are known to cause nerve damage.
Huntington's disease is an inherited neurological repeat expansion disorder, which is one of the most common inherited diseases and can affect people of all races over the world. There is a 50-50 risk of inheriting the disease if a parent is a HD sufferer. Huntington's disease slowly attacks the patient physically, emotionally, and mentally. The carrier becomes aware that they have the disease when between the ages of 30 to 50 years, when the symptoms are no longer dormant unless a genetic test is taken earlier. Huntington's disease causes a slow weakening of the entire body for over 10 to 25 years, which primarily leads to the inability to function and death. It occurs in approximately 1 in 10,000 people on average, and males and females have an equal chance of inheriting the gene from an affected parent. Those who do not inherit the mutated gene will not develop the disease, nor will their children. Huntington's disease does not skip a generation. The symptoms which identify the disease as Huntington's disease become apparent during the ages of around 30 to 50, although in some cases the disease will attack young adolescents or people in their senior years. When Huntington's disease was first discovered, its name was Huntington's chorea, because chorea, which refers to the involuntary movements, was the only known common symptom between all the cases found. It was later apparent that Huntington's disease also affects mental capacity, the ability to organize plans, initiate communication, basic thinking, judgment, and memory, leading to depression and anxiety, personality changes, involuntary movements of the legs, arms, face, and torso, and increased difficulty in walking and swallowing. Mutations which cause Huntington's disease. Damage to the cells in a part of the brain called the basal ganglia is to blame for the lack of control of movement, emotional normality, and cognitive capability in a HD patient. Mutations in the HTT gene cause Huntington's disease. This gene provides instructions for making a protein called Huntington. The function of the protein is unknown, but it appears to play a significant role in the nerve cells of the brain. In all people, the section of genetic material, or DNA, contains a pattern of trinucleotide repeats, which are the building blocks of DNA, consisting of C, G, A, and T. Normally, the CAG segment is repeated 10 to 35 times within the gene, but the Huntington's disease gene was found to contain a specific section that was expanded in the people with the disease. In people with HD, the CAG segment is repeated 36 to 120 times. People with 36 to 40 CAG repeats may or perhaps may not develop the signs and symptoms of HD, but people with more than 40 repeats will most probably develop the disorder. This trinucleotide mutation produces an irregularly long version of the Huntington protein. When the elongated protein is cut into smaller fragments, they become toxic, bind together and accumulate in the neurons, which is troublesome for the normal functions of these cells, causing the abnormal symptoms. By studying a person's DNA and counting the number of CAG repeats, it is possible to tell if that person will develop HD. However, the test cannot predict the age of the onset. Suggested treatments for Huntington's disease. As stated in the Laspada paper, a nagging for the repeat disease field is why the development of the meaningful treatments has not kept pace with the remarkable trajectory of advances in the study of mechanism and pathways. Applying this statement to HD would be very true in this situation. After a 10-year search and the discovery of the HD gene mutation in 1993, much has been learnt about the site of mutation, how certain brain proteins interact with the protein expressed by the HD gene, and how it is detrimental to the human body, but no cure has been found over the last 28-year period. Many scientists are existent in the poignant search for effective treatments to stop or reverse the effects of HD and eventually cure it altogether. Hypothetically, if a HD sufferer or someone at high risk could be administered with an effective amount of an inhibitor of 5-AMP active protein kinase, the polytranslational modification of the gene expression which causes the disease could be suppressed significantly. Although there may be no cure, certain treatments have been successful in suppressing the symptoms. Medications can be effective in treating the common symptoms such as involuntary movement, depression and anxiety, although HD sufferers do better when the medication is kept to a minimum. This is because drugs that are operational at one stage of the Huntington's disease may not be effective at another, and many drugs have side effects. Referral to a physician or a speech therapist is important so that tactics can be put in place for a constructive and long-lasting impact on the quality of life. Nutrition is of high importance to HD patients, as it is essential to consume an extraordinary quantity of calories to maintain body weight, and this can help reduce involuntary movements and other symptoms, particularly in the later stages of Huntington's disease.